Today we're going to go through the stages of mitosis in a little more detail. So first, let me just show you a quick overview of the whole thing. So in interphase, just a reminder, we have our cell. The chromosomes, you can't really see them. They are in this mass called chromatins. They would look like kind of like stringy stuff. You'd have the nucleolus, you have the nucleus, nuclear membrane, and all the cell parts that they don't draw here because they're just sort of saving time. That's interphase. This consists of G1, S, G2. This was your cell cycle. It prepares for mitosis. It does all of its normal jobs, like the cell's doing transport and synthesis and all the things your cell does to keep you alive. So now the cell has decided it's going to divide, and so its first step is going to be called prophase. Now they're showing that there's like a little transition. So what's happening as this cell enters prophase? Well, the chromosomes that were in this tangled mass called chromatin, they're going to condense so that you can actually see them. By condense, I mean they're going to coil up really tightly. So they're going to go from looking like this tangled mass of string to actually looking like these little X shapes like this. Remember, each of these X shapes is one chromosome, and it has two sides. Those are the sister chromatids. And then it's held together, if I change my color here, with this little thing in the middle called the centromere. And that's holding the two sides together. Those two sides are completely identical to each other. A quick reminder from yesterday's lecture that the chromosomes in G1, if you were to coil them up and look at what they look like, they would look kind of like a stick, even though they're not coiled up like that in G1. But in S, they make a copy of themselves so that now they have the two chromatids on the two sides, and now they are ready to move into mitosis. The whole point of that step in S, synthesis, is to make an extra copy of the information. Just like if I had a copy of my notes and I wanted to give you a copy, I would need to photocopy it. I would need two copies so that I could keep one and I could give one to you. So all the information a cell needs is in this one side, but it makes this extra copy of its information so that when the cell divides, each new cell can get a copy. So our first step of mitosis is going to be called prophase. And in prophase, this is where the chromosomes are going to coil up completely. The nucleus is going to disappear. This membrane is going to dissolve. The nucleolus is gone. Remember, this was the nucleolus that its job was to make ribosomes. It disintegrates. And these things start to form. These spindle fibers start to form, and in animal cells, there's these things called centrioles here, and they start moving to opposite sides. So that's prophase. And by the end of prophase, these centrioles are completely at the opposite sides, the spindle is completely formed, and this nuclear membrane would be completely gone. Now they talk about some other stuff here, the kinetochore and stuff. We didn't really get into that much detail. Now, metaphase, these guys are going to line up right down the middle. Now, they're kind of showing them off to the side of one another because they're trying to show you kind of a three-dimensional view of this. But in 2 dimensions, it would look like this. They literally would line up straight down the middle. And, really important, they don't really show it very well here, but these spindle fibers are connected, are literally connected to these centromeres in the middle. That's really important because the spindle fibers are going to, in the next step, are going to start pulling these apart. Well, how are they going to do that? Well, they're going to pull them by these centromeres. So now the chromatids, these 2 sister chromatids, are now going to be called chromosomes on their own. This is going to be anaphase. And these are going to move to opposite poles. They call these 2 sides poles. The middle was the equator, or sometimes it's called the equatorial plate, but the equator's fine. And then in telophase, in the very last step, the chromosomes' new nucleuses or nuclei are going to form in each of the new cells, and the cell typically, the rest of the cell is going to divide. Although that has a special name, it's called cytokinesis. So let's walk through these steps in detail. Now in this particular cell, notice they're drawing it with 4 chromosomes. If this was a human cell, it would have 46 chromosomes. If it was the cell of a fruit fly, it would have 8 chromosomes. If it was from certain species of corn, it would have 300 chromosomes. So every different kind of organism has a different number of chromosomes normally in its cells. Just know, and this is important, that however many chromosomes there are at the beginning, there's going to be the same number at the end. So if this was a human cell, it's going to start with 46, and the 2 daughter cells that are made are both going to have 46. So, prophase, the nucleolus disappears. So there was a nucleolus here, there was like this dark area. It disappears, and the chromosomes coil up. What do I mean by that? Condense or coil up. Remember, before this stage, they were like this stringy mess. The chromosome would look like this. Now, the chromosome coils up super, super, super tight into this sort of X-shaped thing. Oops. Connected in the middle, and that's what they mean by coiling. The centrioles move to opposite sides. What are centrioles? That's these. They look kind of like a little T-shaped thing. And they're going to eventually end up at the 2 sides. They're going to be called poles. Just like we have the North Pole and the South Pole, these are going to be called poles. And this starts to form. This is the spindle fibers starting to build that are going to attach and pull those chromosomes apart. Now, it turns out plant cells don't have centrioles. They do still have an area where the centrioles come from called the centrosome. So in a plant cell, this would be empty in the middle. Spindle fibers begin to form. The nuclear membrane is going to completely disintegrate. And next, we're going to now move into metaphase. So the chromosomes are now, at this point, the spindle fibers go all the way across. Notice the centrioles here. Plant cells would not have these. It would still have this area, but it wouldn't actually have the centrioles. This middle part is called, they're calling it the metaphase plate, but another name for it that we usually use is, it's called the equator. So this is the equator, and these are called the poles. So this is a pole, this is a pole, this is the equator. Again, they're using 4 chromosomes here. If this was a human cell, there would literally be 46 chromosomes lined up in a straight line here. So they line up along the equator. The centromeres are attached to the spindle fibers. So notice, remember, these 2 sides of the chromosome, these chromatids, are technically identical, and they're just hooked together in the middle, and in our next step, they're going to get pulled apart. Again, we're using 4 chromosomes. There would be 46 if this was a human cell. Step 3, anaphase. Okay, so now these chromosomes have started to pull apart, and they are literally going to get pulled to opposite poles. These spindle fibers are actually getting shorter and shorter, and they're pulling the chromosomes towards the 2 sides. So the centromeres are going to split. The chromatids are not called chromatids anymore. Now these are actually called chromosomes. This is what it looks like in an onion cell. This is a plant cell, and you can see them. They're all kind of bunched up, so it almost looks like like 2 little flowers. But these are actually the chromosomes getting pulled towards the opposite poles. And then in our last phase, telophase, the chromosomes are going to uncoil back into chromatin again. We're going to get 2 new nuclear membranes. Keep in mind that these chromosomes are not going to look like X's anymore, right? They're only a stick. They're only like half an X because they had split in half. And usually what's going to happen, it usually starts very, very late in anaphase. Right at the beginning of telophase, cytokinesis occurs. That's the splitting of the cytoplasm. That's where the cells actually split in 2. In animal cells, there are these filaments made out of proteins called actin and myosin, and they literally pinch the cells apart. It's called cleavage, typically. In plants, though, the cells sort of stretch, and this little dotted line forms called the cell plate, and then that sort of solidifies into a brand new cell wall. And so now you have 2 cells. So this step is going to look different in a plant versus an animal, cytokinesis. This is just a little overview. So here's prophase. Here's what they're calling, this is interphase. This is prophase. Here's the nucleus going away. And they're calling this prometaphase because this is like where it's going from prophase to metaphase. You don't have to know the name of this step. It's just the name of the sort of the pathway from prophase to metaphase. But notice they're starting to go towards the middle. Here they are, lined up in the middle. They are connected to the spindle fibers. The spindle fibers, anaphase, are going to pull them apart. And this is an animal cell, so you get what's called a cleavage furrow, which I'll show you a picture of, here. and the two cells are literally going to get pinched apart. In a plant cell, it looks a little different. This forms. This is called a cell plate. It eventually becomes a full-blown cell wall. Checkpoint question. You see an animal cell. You see duplicated chromosomes scattered through the cell. What does duplicated chromosomes mean? It means, so you're looking through the microscope and you're basically seeing this. The chromosomes are X's. They haven't split yet. They're not lined up anywhere. They're just sort of spread out. We don't see a nucleus. So this would have to be prophase. They haven't lined up in the middle. The nuclear membrane is gone. They didn't mention the spindle fibers, but the fact that they mentioned the chromosomes still being X's and the fact that they are duplicated, that's what that means, tells me this has to be prophase. All right. And then cytokinesis in plants versus animals. Again, just a review of what we just said. In plants, you get a cell plate that forms down the middle. In animals, the cells are going to actually pinch apart. All right, so really quick, I'll do a little sketch of everything one more time. So we start with prophase. Prophase, you have the nucleus, you have the nucleolus, and the DNA looks like chromatin. Let's do this one with just 2 chromosomes. We're going to just do this with 2 chromosomes. Now, during interphase, I'm sorry, I said prophase here. During interphase, remember that this would have gone through G1, S, G2. In G1, the cell would have grown. In S, those chromosomes would have gone from looking like this to looking like this, X's, because they would have synthesized DNA or duplicated their DNA. And then in G2, the cell just sort of prepares for mitosis. So this cell is still in interphase, and now it's going to go into prophase. So in prophase, here's what's going to happen. The nucleolus here is going to disappear. The chromosomes are going to start to become visible. I'm just going to do this with 2 chromosomes just to make it really simple. Remember, it just depends what kind of cell it is. The nuclear membrane is dissolving. The centrioles, I'm going to make this an animal cell, so it does have these centrioles. They're like little T-shaped things. They're going to start to move apart, and the spindle fibers are going to start to form. So this is prophase. Now, right before metaphase, so like this is the end of prophase. Here's my 2 chromosomes. Here's my spindle fibers. They're pretty much moved to the opposite sides. So this is beginning metaphase, ending prophase, beginning metaphase. The chromosomes are going to basically move to the middle, which we're going to call the equator. Sometimes it's called the metaphase plate or the equatorial plate, but we're going to call it the equator. Remember, these 2 sides are going to be poles. And then metaphase, it's called metaphase specifically when they are right in the middle. The centromeres of the chromosomes are going to be attached to the spindle fibers. There are some fibers that are not attached to anything. It looks something like that. Here's our centrioles. So this is metaphase. I'll just put an M for metaphase. As soon as these chromosomes split in half, it's now anaphase. So it's going to look like this, and these chromosomes, imagine, are getting pulled. I'm drawing them white so that you could see the centromere attached here. They're actually getting pulled to opposite sides. You still have some of these spindle fibers here, and the chromosomes are moving to opposite sides. And then it's going to be called telophase once they're pretty much done moving to opposite sides. So the chromosomes now, the centrioles are still here. Oops, I somehow made that a highlighter. The centrioles are still here on the ends. The spindle fibers sort of go away. 2 new nuclei, the membranes are going to reform, and the chromosomes, we started with 2, we end still with 2 chromosomes in each cell. It's just that instead of looking like X's, now they look like sticks because they split in half, and they'll go back to looking like X's again when this cell goes through S. And very last step, during telophase, this cell is actually going to split in half. If it's a plant cell, it'll get a cell plate. That's cytokinesis. If it's an animal cell, the 2 cells will pinch apart. But either way, you're going to end up with two cells at the end that are identical to the original parent.